subhanahu wa ta'ala. For this, I request Qari Aziz to recite few verses from Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الرحمن علم القرآن خلق الإنسان علمه البيان الشمس والقمر بحسبان والنجم والشجر يسجدان والسماء رفعها ووضع الميزان ألا تطغوا في الميزان صدق الله العظيم.
Jazakallah. Ladies and gentlemen, the Global Women's Breakfast Series is an event that seeks to celebrate and recognize the accomplishment of women in science and at the same time to build a supportive global network to close the gender gap in science. The GWB is designed to inspire young women and girls to pursue STEM career and to support professional development in early career scientists. ICCBS, under the leadership of Professor Dr. Muhammad Iqbal Chaudhry and Professor Dr. Atta Rahman, is organizing this event regularly since its inception. At the ICCBS, enabling environment is being created for young girls and women scientists. This includes equal opportunities to pursue career in higher education, equal training opportunities abroad in cutting-edge research fields, establishment of daycare centers on the campus, free transportation, monthly stipend to all girls students, flagship program for the training from African women scientists at the ICCBS are few initiatives to begin with. With this, I request Professor Dr. Muhammad Iqbal Chaudhry to say a few words.
Thank you very much, Hina. Good morning and Assalamu alaikum, ladies and gentlemen. I assume that there are several male faculty members are also attending, and this is extremely important. So let me begin by thanking Professor Dr. Mary Garson for initiating this wonderful program, which is, I consider, an extremely inspirational by all definition, because this is truly a day which represents the intellect and highlight the contributions of women scientists. In our country, Pakistan, I often say that this population, 50% of the population is about 90% of intellectual powerhouse. And we're so delighted that, you know, from literally no representation of women in science or in fact in any discipline, the country has actually gone through a major transformation. So we have now 70% of our university population, these bright and brilliant girls who are in literally every discipline of science, engineering, medicines, and they have this tremendous contribution which they are making to the prosperity and well-being of the people of Pakistan. At the political level, we are perhaps in this region the only country which has a representation of about 35% women parliamentarians. So we made sure that their contribution steadily increase. So from the judiciary to media, from academia to industry, there is a growing participation, contribution, and leadership of women, which is extremely heartening. We feel that when we will hand over this country to them, the country would be much better off and will be able to perform much better than what we do today. We are absolutely delighted to have our very dear friend, very inspirational, very respected scientist, Professor Ute Romling, with us. She's a frequent visitor of Pakistan, and she's truly an inspiration for our girls. She has been a great source of strength for us, not only advising our young students, but also giving motivation to our female faculty member, as well as helping our faculty as a whole. So we're delighted to have Ute with us, and her presentation would really be an extremely important one, because that would represent the generic issues, problems which women face all over the world. There are several specific issues, which I'm sure that there would be discussion on it, followed by the presentation of Ute Romling. With this, I would like to welcome you all to this extremely important event. We honestly believe that this is what give us a great source of thinking, critical analysis, and to show us a way forward. We are changing, and we are changing for good. Thank you very much for your participation in this important event.
Thank you so much, sir. And now I request Professor Mary Garrison, if the technical issue is resolved, to say a few words. I'm sorry, Professor, we still cannot hear you.
She's saying that every time she presses it, I guess that this is some problem from our end, because I think the host, this says that host is not allowing. So either you have to create a new link or you have to fix it, Hina, because I think she has been trying to unmute, and every time apparently, as I was getting this message also, that host is not allowing to unmute you.
Professor Mary, can you just unplug your microphone and then do it again? Okay, now we can hear you. Oh, okay, in between when you disconnected, we were able to hear you, but now you just disconnect your microphone.
Yeah, can you hear me now?
Yeah, absolutely. Loud and clear. Sorry about that.
Yeah. No, but I must tell you that there may be a problem at my end, because next to my house there is a building activity and it's very noisy. So of course, but that's too bad, right? So I apologize if there is noise in the background, but it is better that you can hear me than that I speak and nobody hears me, I think. So would you like me to say a few words now?
Yes, please.
Yes? Okay, good. Thank you. Well, look, hello and welcome on behalf of Iupac, distinguished participants, Professor Ute from Europe, I think. Not sure where from, but welcome.
From Sweden, yeah.
From Sweden, welcome. And of course, Professor Iqbal, Dr. Hina, it's just wonderful to meet and greet in this way. Iupac really is enjoying connecting chemists from around the world today, both men and women. Today, I have already had breakfast in Brisbane, then Taipei, then Dhaka, and now with you in Karachi. And shortly I will move on to breakfast in Vienna and Glasgow. I don't think I'm going to manage to make breakfast in North Carolina. But then I return to Mumbai, because in Mumbai, they're having breakfast as late afternoon event. And what we've seen today is a wonderful enthusiasm. Just visualize, everybody, that today, February 16, more than 20,000 chemists, both men and women, are going to be joining in this event. Professor Iqbal will remember that we first ran a breakfast event for the International Year of Chemistry back in 2011, when we celebrated the accomplishments of Marie Curie, Nobel laureate in chemistry and physics. And we had over 100 events in 20 countries. And after that, I said, no, it's too much. I can't do this again. I won't do this again. And then fast forward to 2019, which was the International Year of the Periodic Table. And Dr. Laura from North Carolina, my co-host, my co-chair, wonderful person, suggested to me that we should relaunch the Global Women's Breakfast series. Last Friday, we recorded a welcome video for this event. It goes for less than 5 minutes, and I hope you have the opportunity to play it at some stage. And last Friday, at that point in time, we had over 300 breakfasts in over 60 countries joining in. To my complete amazement, when I woke up this morning and I checked my phone, I discovered that we now had 403 breakfasts in 78, possibly 79 different countries joining in. So in the last week, since last Friday, which is 5 days, over 100 different groups worldwide in more than 20 different countries have joined in. And this year we have 9 new countries who've never participated before. So just want to say a few words about the purpose of the breakfast, which is very much, it is to celebrate women in chemistry, but it's also, in my opinion, to celebrate young people in chemistry, because young people are going to be the future of chemistry, of science. And if we think about, in particular, about next year, when UNESCO is launching, well, in fact, it's launching it later this year, an International Year in Basic Science and Sustainable Development, I took a little bit of time to warm up to the idea of yet another international year that Iupac had to be involved in, because we've had 2 that I've been involved in. But when I thought about it, I realized that your group there in Karachi at the International Center for Chemical and Biological Sciences and at the University of Karachi, to be quality researchers, which you are, one of the skill sets you have to develop is to think critically about your own science and also about the science that other people around you that are doing, because you have to be able to tell when somebody else's science is perhaps not as good as it should be. That's correct, this is not correct. You have to make those sorts of decisions. So you have to think critically about your research. To do that, you've got to have a good store of knowledge, of scientific knowledge up here. And where does that good store of scientific knowledge come from? It comes from an excellent grounding in the basic sciences. If you are able later on to look at the welcome video, you'll see a little clip where I comment that when I was an undergraduate student in England at the University of Cambridge many, many years ago, I never had any idea that I would end up in Australia, scuba diving on the Great Barrier Reef to collect marine samples and being able to call it work. I mean, how wonderful to be able to do something like that. But I had no idea when I was a young student exactly where I would want to end up. So that's actually going to be the first message that I want to share with you today. Although I'm aiming these messages at the young people, I think that we, the senior people in the audience and listening globally, we, in our role as leaders in the chemical discipline, we need to ensure that these messages get heard and that we consider them to be good practice also. So what I was talking about just then when I commented about as an undergraduate, I never had any idea that I would end up in Australia. And I certainly had no idea that I would create an event which leads to my contacting scientists in 78 different countries. So this is actually about cultivating your skill set, your hard scientific skills, but also your soft skills, communication, social media, that provide you with the opportunities to develop your future careers and your future professional development, because you are the future of chemistry. We've got to think about how chemistry will impact on science and on sustainable development and, of course, on global health. And how important is that now as we are currently still in this dreadful pandemic? So cultivate your skill set, everybody. And your teachers and mentors are exactly the people who should be guiding you through how you can develop. I also want to comment that actually every single one of you are important. That means that you need to think about your work-life balance, and you need to think about your mental health and your physical health. It's important to maintain a good equilibrium. You really can't do chemistry research if you are completely exhausted or not mentally in the correct loop. So don't underestimate how important it is simply to consider that you are important. So I think I was talking about opportunity, and I think it's valuable to think about external opportunities. And Professor Iqbal has commented about your collaboration with Professor Utter. And so that sort of interaction with scientists from overseas represents an opportunity, an external opportunity. And it's an opportunity to promote yourself, your science, and of course, your visibility. And also the visibility of the people around you, because let's not forget that chemistry and science is actually a team activity. One of the key reasons why we're highlighting diversity and inclusiveness is because the more diverse teams, and you have these in Karachi, I know that, they are the more creative teams, and they are also the happier teams. There is a better balance in your work and professional lives if people are happy and also if they are creative. And that comes from diversity, comes from acknowledging being part of a team. So it's not just your visibility, it's the visibility of everybody around you. So in particular, for the senior people in the field, that means looking out for opportunities to put young people forward for job opportunities or awards and so on. And also to encourage the young people with you to consider joining what I like to think of as young Iupac, which is the International Young Chemists Network, which I think is quite strongly based in Europe and North America and would benefit from having more representation from Asia. And I'm happy to put you in contact with people there. As I said, I think it's young Iupac, and it is the future of the science. I think just one final message, and I pick my words carefully here. It's about speaking up and speaking out. It's being prepared to comment when things don't seem quite right. Regrettably, there are still institutions and organizations worldwide where the academic culture is not quite as we would like, where academic practices are not transparent, funding issues may be not transparent, job opportunities may not be fully transparent. And collectively, these are the sorts of things that we just need to overcome, those sorts of barriers, so that there is an equity, everybody has an opportunity to progress in the way that they would like. So for the young people in the audience, I hope that this is going to encourage you to continue as chemists and the allied disciplines. We need you. And of course, for the senior people in the audience as leaders, and I certainly, and I know that the people I'm speaking with now, we offer to help you and to work with you to achieve your potential. I hope that those few words have been helpful to you. I'm very much interested to listen to some of the discussion that follows. I will have to leave you eventually because there is another event that I promised that I would join, but it's certainly for a little while I can stay with you. So congratulations everybody for organising your event there in Karachi. Please do use social media to connect with the rest of the Iupac world and let us know about your event. And thank you very much for inviting me to join you.
Thank you so much, Professor Mary, for such enlightening remarks. With this, I now request Professor Dr. Otte Romling, Department of Microbiology, Tumor and Cell Biology, Biomedical Karolinska Institute, Stockholm, Sweden, for her talk. The title of her talk is Why I Became a Scientist, Challenges and Rewards. Over to you, Professor Uday.
Can you please let me share my screen?
Yes. Have you shared your screen, Professor Uday? We cannot share it.
I cannot share my screen. Now I think I can. Yeah, first of all, I would like to thank Professor Chowdhury and everybody, all the hosts, for inviting me to give a lecture at this occasion. I feel very honored to do that. I would like to give a very personal view on science, and also I would like to encourage you to carry on in science. Also, you think maybe I don't know so much, I'm not maybe as good as others, but all those obstacles usually can be overcome if one is working hard and is persistent and believes in oneself. And I would like to give you such an example because I, in some way, experienced this also by myself. So this will be a very personal guide through my very first build up of a scientific career, but that was then also the foundation for my future scientific career. Today, I'm a professor at medical microbial physiology at the Karolinska Institute. And here you see the Aula Medica of our institute. And in this building I am working, it's a new building called Biomedicum, very pleasant atmosphere in this building and very new laboratories. So this is my professional curriculum vitae. Today, I'm professor at Karolinska Institute, and my career seems to be very straightforward, from studying biochemistry to PhD to postdoctoral work, being a junior research group leader and associate professor. However, during this time, I had to face very many obstacles and I have to solve very many problems, but I also got some rewards for that. So my major research interests for a long time have been the mechanisms of worldwide spread of Pseudomonas aeruginosa clone C, and the biofilm formation in Salmonella Typhimurium and its regulation by the novel second messenger cyclic di-GMP. And I would like to tell you how I could build up those two research projects. I used to say it was almost all was serendipity. However, one can also say fortes fortuna adiuvat. The luck is with the brave. So at the beginning of my scientific career, I graduated from secondary school in Germany, and that was a musical high school. So I was basically not exposed to life sciences, and I also had not much idea about life sciences and about the study of biochemistry that I choose. So I was very surprised in the very first run that we had long laboratories in the afternoon during the studies and not only lectures. However, I mastered this, and then when it came to do my master thesis in biochemistry, I was faced with a question how to choose the first research topic. Something to be interested in, but it looks like everything or many topics are interesting. So how do I know what I'm really interested in and what I can do? I went to a researcher in my institute who was known as a young research group leader to do interesting work. And he gave me two possibilities, either to map the Pseudomonas aeruginosa PAO genome or to isolate mRNA from cystic fibrosis patients. I then chose just a little bit, basically serendipitously, to map the Pseudomonas aeruginosa PAO genome. And why did I do that? Maybe because sometime previously I gave a presentation about a physical map of the Escherichia coli K12 genome, and I found that quite interesting. Physical map is the placement of restriction sites on the on a bacterial genome, and this was possible at that time with a technique that is called pulse field gel electrophoresis. That was just invented 5 years earlier by Schwarz and Cantor in 1984. Pulse field gel electrophoresis enables the separation of large DNA fragments on a gel by an alternating electric field. And with this technique, fragments up to megabase pairs in size can be separated. There has been several, after the initial invention, there has been several technical advances in this field, and I use simply one of them, which is called Chef gel electrophoresis. I was fortunate in the laboratory where I have been that there was put forward an idea how to map the restriction site on a bacterial genome in a very fast way. And that was by two-dimensional gel electrophoresis, where in the first dimension, a partial digest was performed, and in the second dimension, a total digest with a restriction enzyme was performed. And by analyzing the pattern of DNA fragments that arise after the second dimension, one can, like in a puzzle, construct the macro restriction of a bacterial genome. I did that in my master's thesis, and I was very fortunate to obtain the macro restriction map of the Pseudomonas aeruginosa PAO genome. We could publish that in a very prestigious journal, in the EMBO journal in 1989, and then a substantially improved map also in Nucleic Acid Research in 1991. This macro restriction map actually helped also to solve the sequencing obstacles upon de novo sequencing of the Pseudomonas aeruginosa PAO genome. And since this technique is very fast, we were then able to map not only Pseudomonas aeruginosa PAO, we also mapped other Pseudomonas aeruginosa strains, Neisseria gonorrhoeae, and Pseudomonas stutzeri. So since this project had been quite successful at the end and resulted in a master thesis called a macro restriction map of the Pseudomonas aeruginosa PAO genome, I carried on in my PhD and later on also in my first postdoc on this topic in the same research group. And what I can only tell about the experience after my or during my PhD and also master thesis and the postdoctoral work is try always to do your best, but also go against the convention if needed. And very important, trust your own results, but only with the right controls. So what does it mean? I will give an example from my PhD thesis. So the macro restriction fingerprint pattern is a high resolution typing technique. What is displayed here on this gel with the restriction fragments are different Pseudomonas aeruginosa genomes. And these genomes have all different macro restriction fingerprint patterns, which means they are genetically diverse. But when those genetic diversity results in a genetic similarity, one has a similar strain, or similar strains are then be put in a group that is called a clone. This technique can then be used to answer questions in clinical microbiology, like what is the source of the infection? Does transmission of strains occur between patients? And what is the evolution of strains, for example, during an infection? But to answer this question, one has to define the criteria. What are closely related strains? How many bands need to be similar, and how many bands on this macro restriction fingerprint pattern have to be different to be still belonging to closely related strains, basically to a clone. So what we did during this work, we analyzed more than 600 Pseudomonas aeruginosa strains, and we could come up with a definition of a clone. We defined clones based on the epidemiology and based on the similarity of the macro restriction patterns as macro restriction fingerprints patterns that are different up to 6 bands. This was going against the convention because the convention defined the differences of 1 or 2 fragments as an indicator that those strains belong to a clone. However, the Pseudomonas aeruginosa genome is very large and also very diverse. So we held up to this definition, despite that other researchers were not accepting this definition. Using this definition, we could show that 30% of the isolates isolated from cystic fibrosis patients in Hanover, Germany, belong to a clone. And we could also, for the first time, identify clone C strains in the environment and in the aquatic habitat. So with this definition also, we could identify one clone that was named by us clone C as highly prevalent. We could publish these findings, the definition, in 2 I would say, benchmark papers, one published in Applied and Environmental Microbiology, and the other publication in Journal of Infectious Diseases, which described the epidemiology in cystic fibrosis patients. Clone C has several unique characteristics. Clone C harbors 600 kilobase per more DNA compared to the reference strain PA01. So it has much more genetic possibilities. And on the other hand, we could also show that cystic fibrosis patients, when infected with Pseudomonas aeruginosa clone C strains, the clone C strains evolve and display frequently large chromosomal inversions, which are a specific adaptation mechanism to the cystic fibrosis lung habitat. And these large chromosomal inversions do not occur only by chance. They basically occur through transposition of insertion elements into genes that are that have been shown to be important for the adaptation of Pseudomonas aeruginosa to the cystic fibrosis lung environment, and then lead to rearrangements of the chromosome. based on this work during my early time, during my PhD and also postdoctoral work, I have not left Pseudomonas aeruginosa clone C. We still work with Pseudomonas aeruginosa clone C strains. Clone C has been identified as one of the most prevalent Pseudomonas aeruginosa clones. And to elucidate the molecular mechanism of worldwide spread, this is our mission at the moment. We can imagine several mechanisms of success, like the features on the core genome, features on the accessory genome, or a combination of both features can lead to the spread of this clone and of its clonal members. And to answer this question, we have decided to focus on the so-called transmissible locus of stress tolerance of Pseudomonas aeruginosa clone C. This transmissible locus of stress tolerance is up to 20 kilobase pair genomic island that is basically a core genome feature of clone C strains. This genomic island encodes for gene involved in protein homeostasis, and actually as a hallmark, those horizontally transferred genes or gene products are usually or often homologs of core genome genes. So we have not only one copy of those genes on the genome, but we have two distinct copies on the genome through the transmissible locus of stress tolerance. This shows that distantly related homologs can potentially widen the substrate spectrum and the metabolic abilities of strains and are not just cargo genes. Another obstacle in Pseudomonas aeruginosa research is biofilm formation. Biofilm formation is a hallmark of Pseudomonas aeruginosa and also contributes to acute and chronic infections. We have had many model systems for biofilm formation, but what we observed is that all those models maybe not always resemble growth conditions in the host. So at one point I decided to look at this aspect and try to come closer to growth conditions in the host. The developmental cycle of a biofilm starts from a single cell that is sensing a surface and then eventually develops into a mature multicellular structure with the production of extracellular matrix components. This is what is displayed here. The surface grown biofilm is basically the paradigm of biofilm formation and has been studied in Pseudomonas aeruginosa equally as in other bacteria. Those surface attached biofilms are tolerant against antibiotics and the immune system with various mechanisms. And here you can see a Pseudomonas aeruginosa biofilm on a surface in reality. And you can maybe appreciate the mushroom-like structures and the multicellularity of basically single autonomous organisms. However, this surface-grown biofilm does not really reflect the biofilm formation in the cystic fibrosis lung, where Pseudomonas aeruginosa is not attached to the epithelial cells, but resides in aggregates in the mucus. So what we did is we developed a so-called artificial sputum medium, which resembles the nutrient composition in the cystic fibrosis lung environment. And using this artificial sputum medium, we could indeed observe cell aggregates as biofilms, and Pseudomonas aeruginosa in this model did not adhere to the wall of the well of a abiotic well in the culture tube. We could also observe additional hallmarks of biofilm formation and of Pseudomonas aeruginosa chronic infections, like the mucoid conversion within 48 hours, the small tight aggregates, biofilm-like, that resembled cell aggregates in the cystic fibrosis lung, the reduced motility of bacteria, and the increased impression of outer membrane proteins. So this novel model of biofilm formation of Pseudomonas aeruginosa in the cystic fibrosis lung has been widely accepted in the scientific community. As a second part of the lecture, I would like to go to the second topic that occupied me for a long time, and that is cyclic di-GMP. And this is basically a history of rediscovery. After working for 7 years on Pseudomonas aeruginosa during my master thesis, my first PhD, and my first postdoc, despite of working in a very successful and highly inspiring environment, I decided I need a change. And I decided to do a postdoctoral work at the Karolinska Institute. It turned out that at the end, I worked on biofilm. I worked on a project that we now know is biofilm formation in Salmonella Typhimurium, but at that time it was not known. Also, when I started to write my application, I was actually suggested to work on antimicrobial resistance, but we spontaneously decided to work on another project. And this project is shown here. These are 2 strains of Salmonella Typhimurium. On the left side, you see a standard Salmonella Typhimurium colony, and on the right side, you see a mutant Salmonella Typhimurium colony that we later called the rdar morphotype. And the rdar morphotype stands for red, dry, and rough. And we could define this colony morphology as a multicellular biofilm behavior. But in the first run, the questions that I posed were very much more basic, and that was, what is the difference between the 2 strains and what is their role in virulence? And I faced a major obstacle, actually. I could not readily answer the first question because the strain pair that I got was not susceptible to genetic manipulation. And at that time, when I started this work, sequencing was not an option because it had not been developed in that way that one could readily sequence bacterial genomes like we can do today. So what should I do? I could leave this project, or I could look for a solution. And I decided I looked for a solution, and I made my chance. And the solution was I ordered from the ATCC strain collection another strain, and I was hoping that I find a similar phenotype like the one that I had displayed on the slide before. And indeed, with a frequency of around 10 to the -2, 10 to the -3, I did find those rdar colonies also in the seemingly pure ATCC 14028 culture collection. So I could continue with my work as the ATCC 14028 strain was susceptible to genetic manipulation. So what we then during this a little bit more than 2-year postdoc found out is that this rdar morphotype on a agar plate grown in a very simple way is regulated by a transcriptional regulator that is called CSGD. And when this transcriptional regulator is deleted, we see a colony morphology that does not display any multicellular behavior and not any rdar morphotype. So we could publish this work then in the Journal of Bacteriology and in Molecular Microbiology, 2 very reputed journals. And these were actually very basic research findings that inspired not only us, but also inspired many other people. So what were the sources of the inspirement? We could show in this work that we could define a multicellular biofilm behavior in Salmonella Typhimurium, and that has not been shown before. We could then identify the major activator of this rdar biofilm formation, the transcriptional activator CSGD. And we could also identify amyloid curli fimbriae as part of this extracellular matrix of this rdar biofilm. However, when I deleted the curli fimbriae genes, I did not produce a SAW morphotype, but I produced this type pink morphotype. So that indicated to me is there need to be other genes that are regulated by the biofilm regulator CSGD. And also, what is the identity of this other extracellular matrix component that binds the di-GMP that is integrated into the agar plate and results in such a pink colony morphology. We can distinguish, this is just to distinguish between these 2 extracellular matrix components. So we have on the upper panel, the unknown, at this time, unknown extracellular matrix component, and on the lower panel, we have the curli fimbriae expressing strain. So we could see that 2 distinct biofilm phenotypes are expressed by the 2 different extracellular matrix components. So how to find out what is the other extracellular matrix component? If you have something unknown, of course, you can first try to find or to do assays for known components. I did those assays for known components, and I could not find any indications what is this extracellular matrix component that binds the di-GMP. But I would like to introduce you now to the power of an unbiased screen by transposon mutagenesis. So when you don't know what you're looking for, you can only look unbiased to get a rational conclusion. And I did this unbiased screen by transposon mutagenesis. And what we found is we identified what we now know as the cellulose biosynthesis operon in Salmonella Typhimurium and also in Escherichia coli, because we had transposon insertions in the cellulose synthase BCSA and then alongside the entire cellulose biosynthesis operon. But at that time, it was not possible to, or it was not a common knowledge that a cellulose biosynthesis-like operon is indeed producing cellulose. So besides the genetic evidence, we also needed a chemical evidence. Cellulose is actually a linear 1,3-beta-D-glucan composed of the disaccharide cellobiose. And that leads to linear glucan chains that rearrange in parallel to each other, and in that way form a crystalline structure that is very hard to tackle by chemical agents. And when you use very harsh conditions like acetic acid, nitric acid, you can see that those colonies which take up the Congo red with a pink color have only glucose left, and the colonies that are white and do not display an extracellular matrix do not have any sugar detected. Linkage analysis then showed us we have also a 1,4 beta-glucan, and that showed us that indeed that the genetic evidence was true. We have basically a 1,4 beta-D-glucan and cellulose as an extracellular matrix of Salmonella Typhimurium, in Salmonella Typhimurium and Escherichia coli rada morphotype. This research we could publish in Molecular Microbiology in 2001, and that was basically my first my first doctoral student, Savit Sogai, who did this work. And that work also caused very much excitement in the scientific community, because it was shown for the first time that Escherichia coli and Salmonella Typhimurium produce cellulose outside of 2 model bacteria. And it also was a starting point for a more systematic investigation of extracellular matrix components in bacteria by the scientific community. Previously to our work, only 1 gram positive bacterium, Staphylococcus epidermidis, has been investigated in such a way. So, but today we know that many organisms produce cellulose, and also many microbes and bacteria produce cellulose. So we have not only the plants and the animals, amoeba and the social amoeba, we have many bacterial species throughout the phylogenetic tree, and not only Salmonella Typhimurium, which actually produce cellulose or a variant of the cellulose macromolecule. But we also, after detecting the cellulose, we knew that we have more genes that are involved in the other morphotype. We don't know how is this morphotype regulated. So to detect CSGD regulated genes, we decided to do a more sophisticated assay, and we use CSGD itself and turn CSGD on and off in order to find more genes involved in the rdar biofilm morphotype. So to do that, CSGD was placed on a plasmid with a tight inducible promoter. And then we performed transposon mutagenesis with a transposon which has an integrated reporter gene. And then we differentially induced CSGD expression with the inducer aeruginos. So on the left side, you see the agar plate without aeruginos, and we see colonies that have the reporter gene on and the reporter gene off, because they randomly integrate into the chromosome. On the right side, we turned CSGD on in the very same colonies. And you can see that, again, we have this display of colonies that are on and colonies that are off. But you can also see 2 red colonies that are on in the aeruginos positive plate and not on in the aeruginos negative plate. And those 2 colonies are specifically contain genes that are specifically turned on upon turning on CSGD expression. And we analyzed then those two genes or colonies, more colonies, and what we could find is I could find integration of the transposon in basically one gene. And this gene turned out to encode for a protein that has a so-called GGDEF domain. And we called this gene or gene product ADRA. We knew from the genetic study that it regulates cellulose biosynthesis in Salmonella Typhimurium. And at that time also we could see that we have in the database other gene products which actually have a hallmark of the ADRA, namely the C-terminal GGDEF domain. I would like to draw your attention to the first two proteins, which are a phosphodiesterase and a digranulate cyclase, and those are from the cellulose-producing bacterium Gluconacetobacter or Comagataebacter xylinus. We could publish this work again in molecular microbiology, showing that the transcriptional regulator CSGD, which at that time we had called HEFD, is a checkpoint of multicellular and aggregative behavior and regulates not only the curli fimbriae, but also a gene that is encoding for a GGDEF domain protein. I would like to emphasize here that the experimental design is very important, and sometimes very subtle changes in experimental design can give you different results. What you can see here in this graph is the transcription activity of a fusion to the cellulose biosynthesis operon genes, to the cellulose synthase BCSA, and this last gene in the operon BCSC. And these genes are indeed not regulated by CSGD. So with this sophisticated differential expression of CSGD, the cellulose biosynthesis operon would have never been detected because it is not regulated by CSGD on the transcriptional level, and also not by other hallmarks of the radar morphotype, the RPOS gene, a sigma factor, and also not by ADRA. So ADRA definitely is not a transcription factor and is not affecting transcription. So our next question was then, how does ADRA activate cellulose biosynthesis? And I have to go back to Gluconacetobacter xylinus with the GGDEF EAL domain proteins that I have shown you in the previous slide. At that time, when this work was started, GGDEF and EAL was not a common term. The GGDEF domain was called domain of unknown function 1, and the EAL domain was called domain of unknown function 2. As you see, domain of unknown function, because it was not known what they are concretely doing. And domain 1 and domain 2 indicates those protein domains are actually one of the most abundant protein domains in bacterial genomes. But what was known at that time already is that those two proteins which contain the domain of unknown function 1 and domain of unknown function 2 are actually involved in the synthesis and hydrolysis of a molecule that is called cyclic di-GMP. And this molecule has been shown to regulate cellulose biosynthesis in Gluconacetabacter xylenus. What has been cyclic di-GMP? There has been very scarce reports in the literature about this molecule. What we knew at the time only that it was found in 1987 in the search for an allosteric activator of the cellulose synthase in Gluconacetabacter xylenus, a fruit-degrading and soil bacterium, and that led to the identification of cyclic di-GMP. This finding was based on the previous observation of a discrepancy between in vivo and in vitro cellulose biosynthesis. In vitro, a soluble allosteric activator was missing. And it was known that the allosteric activator is a GTP derivative. And in a more than a decade-long search for this activator, eventually cyclic di-GMP had been identified by the group of Moshe Benzeman. So this is the cyclic di-GMP second messenger. It is a 3' connected GMP guanosine monophosphate. So how to demonstrate then in our model system in Salmonella Typhimurium what the GGDEF and EAL domains are doing, and in this case, what is our protein ADRA doing? We could put forward the hypothesis that the GGDEF domain, the domain of unknown function 1, is actually producing the cyclic di-GMP based on our previous genetic phenotypes and evidence. However, we should rigorously also show it. So again, we used our very simple agar plate model system in order to demonstrate that. So we expressed ADRA in Salmonella Typhimurium. And when we express ADRA in Salmonella Typhimurium, we indeed get production of cellulose biosynthesis, while in the control strain, we have no cellulose production. This cellulose production is associated with a rise in the cyclic di-GMP production, as demonstrated by HPLC, high pressure, and then using this technique, we saw a high peak of cyclic di-GMP that corresponded to the chemically synthesized cyclic di-GMP, while in the control strain, not such a peak has been observed. The mass spectrometry confirmed the molecular weight of the cyclic di-GMP, which is 689 Dalton, and the electrospray mass spectrometry by fragmentation confirmed the identity of the compound. So then what about the EAL domain? The EAL domain, based on our genetic, initial genetic evidence, should be then the phosphodiesterase that is degrading cyclic di-GMP. To show that, we used again the agar colony assay, and we expressed ADRA and get cellulose biosynthesis, but upon co-expression of a phosphodiesterase that is YHJH, which contains only the DUF2 domain, the EAL domain, we could show that co-expression of this EAL domain is counteracting cellulose biosynthesis. So we concluded that cyclic di-GMP must be degraded in this construct. And indeed, when we expressed ADRA in a concentration-dependent way and keep YHJH, the phosphodiesterase, constant, we can see that we dramatically reduce cyclic di-GMP synthesis in contrast to the strain where only ADRA is expressed. That is shown with the dotted line. We further then observed that ADRA and YHH, the cyclic di-GMP synthase, and the cyclic di-GMP hydrolase is not only regulating the cellulose biosynthesis, it is also regulating another fundamental behavior of bacteria, and that is motility. So in this slide, you can see that, that you can see that upon overexpression of the ADRA, the GGDEF domain protein, which is shown as 2, you can see a reduction of swimming and swarming motility. And upon overexpression of YHGH, we can see a significantly enhanced behavior of swimming and swarming. And not only flagella-mediated swimming and swarming, also twitching motility, which is based on type 4 villi in Pseudomonas aeruginosa, is inversely regulated by GGDEF and EAL domain proteins. So compared to the wild type vector control, expression of ADRA leads to a downregulation of twitching motility, while expression of the phosphodiesterase YHGH leads to an upregulation and enhanced twitching motility behavior. So based on these very simple plate assays, actually, we came forward with a hypothesis not only for Salmonella Typhimurium and Escherichia coli, but with a hypothesis that we at that time thought is valid maybe for other bacteria as well, as we observed this behavior also in Pseudomonas aeruginosa. And today it has been found that actually this model that we have put forward is valid not only for these two organisms, it's valid for any bacteria where it has been investigated. So this model says that when we have high cyclic di-GMP second messenger, we promote sessility of the cells on the single cell level. And this sessility is directly associated with biofilm formation and with the production of adhesive extracellular matrix components like cellulose and amyloid curli fimbriae. On the other hand, when we have low cyclic di-GMP level and cyclic di-GMP is hydrolyzed by the EAL domains, the phosphodiesterases, we promote motility on the single cell level. And not only twitching and swarming motility and swimming motility, today it has been found that also many other motility behaviors are actually regulated by low cyclic di-GMP levels. We could publish this work again with Roger Sim as the first author, another PhD student of mine, actually my first PhD student after I had moved back to the Karolinska Institute and started my position as an associate professor in molecular microbiology again. And this work was an inspiration for not only for us, but also for other people working in molecular microbiology. And this sessility motility transition between that cyclic di-GMP is conferring was demonstrated in many gram-positive and gram-negative bacteria. So what makes cyclic di-GMP so exciting, besides that it regulates a very fundamental lifestyle transition in bacteria? The other excitement in the scientific community and for us is the complexity of the cyclic di-GMP network. We can have in some bacteria more than 100 cyclic di-GMP turnover proteins. And in Salmonella Typhimurium, with 20 cyclic di-GMP per approximately 20 cyclic di-GMP turnover proteins, we have, as you can compare, a relatively modest number of cyclic di-GMP turnover proteins. But a high number of them, actually more than 8 of them, already regulate the biofilm regulator CSGD. So CSGD does not only regulate itself, the digranulate cyclase and GGDEF domain protein ADRA, which is leading to cellulose biosynthesis. CSGD is also regulated by several cyclic di-GMP turnover proteins, positively and negatively. And with respect to the cyclic di-GMP network within regulation of motility, this regulation of motility is not simply a switch on and off of the functionality. Cyclic di-GMP signaling affects also the flagellar rotation, direction, and speed. The cellulose biosynthesis, which is affecting motility, but also secretion of monomeric flagellin and the amount of cell-associated flagellin in order to promote biofilm formation under certain circumstances. So this is a very complex regulatory circuit that has to be sorted out for each and every phenotype. But the diversity of the functionality of cyclic di-GMP signaling just in this simple flagellar mediated motility is very exciting. But besides biofilm formation, cyclic di-GMP does much more also in Salmonella Typhimurium. And the hallmark of Salmonella Typhimurium is the invasion of epithelial cells, which is leading to virulence. And this invasion of epithelial cells is preceded by the contact of epithelial cells, which is basically resembling the first steps in biofilm formation. But indeed, and also this biofilm resembling approaching of epithelial cells is regulating by cyclic di-GMP. And this is just an example to show that several digranulate cyclases encoded by the Salmonella Typhimurium genome and several phosphodiesterases encoded by the bacterial genome are actually regulating this invasive phenotype. Cyclic di-GMP signaling affects, however, not only invasion, it affects also other virulence associated phenotype, like the induction of the pro-inflammatory cytokine IL-8. And surprisingly, in all of these phenotypes, also the biofilm activator CSGD plays a determinative role. So in summary, we have come up then with a model system of cyclic di-GMP regulating not only biofilm formation, but also virulence of Salmonella Typhimurium. And that occurs through partly through the biofilm regulator CSGD, partly through cellulose biosynthesis, but also most likely through direct regulation of, for example, type 3 secretion systems. Excuse me. I would now just like to give a more global picture of the cyclic di-GMP signaling. When we started this work with our first publication in 2004, this was a hallmark of the cyclic di-GMP signaling research. However, this research was very readily diversified by many other researchers and contributing to the different aspects of cyclic di-GMP signaling affecting physiology. And this, since cyclic di-GMP signaling is not only present in Salmonella Typhimurium, but also in many other bacteria, cyclic di-GMP is also a so-called pump or mump molecule, microbial associated molecular pattern, a conserved pattern that is recognized by the innate immune systems of higher organisms. So cyclic di-GMP regulates the transition between sessility and motility in bacteria, and it regulates the transition between commensalism and virulence, but it also can prevent extracellular biofilm formation, actually a phenotype that has not been followed up so far, but it was an initial observation by researchers. Besides these microbial-based physiology, cyclic di-GMP has been found to inhibit growth of cancer cells, and it's as I stated already, it's a microbial-associated molecular pattern. That means it's recognized by the immune system of higher organisms, and it's also in discussion as a non-cytotoxic adjuvant. So cyclic di-GMP is an interkingdom crosstalk molecule, and it might contribute to cancer treatment through its effect on biofilm formation and inhibition of cancer cell growth and immune stimulation to overcome immunosuppression in the cancer environment. But cyclic di-GMP is not the only answer. We know today that we have many more bacterial cyclic dinucleotides, and I list here only the most predominant ones, which besides cyclic di-GMP are cyclic di-AMP and cyclic di-AMP-GMP. And in 2013, also a eukaryotic cyclic dinucleotide had been discovered, cyclic di-GMP-AMP, which is actually at the heart of innate immunity in higher organisms, including humans. So the role of cyclic di-GMP in innate immunity is as new adjuvants, and with respect to sensing of cyclic di-GMP, there is a heterogeneity in susceptibility to bacterial cyclic dinucleotides in the human population. And that can lead to susceptibility to various infectious diseases, and also to the nutrient and metabolic status that affect human mortality. Cyclic di-GMP is also involved in DNA-specific or cyclic dinucleotide sensing is also involved in DNA-specific autoimmune diseases such as systemic lupus erythematosus. In the bacterial world, the impact of the cyclic di-GMP second messenger does not only relate to biofilm formation and motility and virulence. It has been shown that cyclic di-GMP is also involved in the biogeochemical cycle of organisms, in the global climate, in antibiotic production, in photosynthesis, in radiation resistance, and in corrosion, and certainly in many other, it regulates very many other phenotypes that I have not listed here. Last but not least, I would like to thank all my previous PhD students, postdoctoral workers, and other colleagues that worked with me, and all my collaborators for this making this work possible. In particular, I would like to thank my PhD supervisor, Burkhard Tümmler, and Professor Maas. I would like to thank my postdoctoral supervisor, Staffan Normark, and also I would like to especially thank Iqbal Chowdhury and Humaira Jahan, who invited me kindly to visit the University of Karachi, and we collaborated for a long time. And you, I would like to thank you for listening.
Thank you so much, Professor Ute, for this journey which you shared with us, largely your scientific career. We do have a couple of questions in the chat box. Professor Summer from our center, she has asked, like, kindly do let us know about the hurdles or challenges faced by you to secure research funding as a woman scientist. Professor Ute, are you with us?
Yes, yes. I would like to say that sometimes I face challenges to secure research funding. Also, I face challenges that with questioning the autonomy. Like maybe not as much today, but previously, yes. For example, when I had friends and friends of the other gender, it was often the merits were imposed to them. So so I was not. I was, and I'm also, I would say, personally, I'm relatively shy. So I maybe don't approach people in the first instance. I take contacts cautiously, and that is interpreted as not competent. So you have to be, as a scientist, maybe always very direct and very outreached to be accepted.
Okay.
I'm not sure that this is really a gender bias, but I would say that maybe women are not really, sometimes they are maybe not so sure about their capabilities for themselves, or they don't value their capabilities in the same way as males do.
Mm-hmm. We do have another question. Have you faced any discrimination throughout your career being a woman scientist? If yes, how did you handle that kind of discrimination? Whether it comes to like promotion or training opportunity or something.
Well, I like or I prefer to focus on the scientific questions.
Okay.
I think we can never. Of course, there are many different factors which maybe affect promotion or affect visibility. But I would say the advice that I would give is try to do solid work. Of course, maybe not only solid, but a work solidly based in scientific evidence and in the knowledge which is already present, and then advance this knowledge when you have the opportunity. And those opportunities are maybe much more often there than not. But one of the scientific capabilities, and also what one has to learn in science, is to recognize when these possibilities, so to say, come to you through experiments.
Right. Thank you. And last question, as a woman scientist, how you were able to manage professional and personal life? How did you balance both life?
Well, I would say that is always a challenge. and one has to try to do the best. I have a son, and I hope we have raised him well. Fortunately, in Sweden, we have many opportunities of also childcare, and also we have opportunities that women who have children and work in science are not considered as bad or as mothers who neglect their children. In some other cultures, that is not other cultures are culturally different, and working women are not readily accepted. In Sweden, I would say, also due to the entire society structure, working women are accepted more than in other countries, I would say. But also, I think also other countries have improved in this in the last 20 years.
Thank you so much, Professor Ute. With this, I would like to thank you for your time and discussion. Professor Mary is here with us again. Professor Mary, would you like to add some concluding remarks? Your mic is mute.
Hopefully you can hear me now.
Yeah.
Okay, good. Thank you all. I'd really like to thank Professor Ute for her fascinating talk. Because of course, although it was a molecular microbiology and there was a lot of chemistry in it, so she illustrated very well that link between the disciplines, which is the hallmark of excellent science. And also, I think the messages that she gave us about perseverance, the willingness to change country, and to reflect on how her career was progressing. I think those are important messages for your audience. And of course, she talked about opportunity and visibility. And so that key message about be visible through your scientific research and your scientific evidence. That for me was, so thank you very much, Professor Ute, for your talk. And I do hope that we get the opportunity to meet in person at some point in the future. Thank you to everybody in Karachi for joining us today. I hope that you have done some social media, or if not, that you will do some. I took a couple of images. I hope it's okay that I post those on Twitter. I know some people are sensitive about photographs, but we want to show the diversity of events. And we look forward, we hope that you will join us this time next year when we celebrate yet another, the 5th in the series of Global Women's Breakfasts. And thank you particularly, Dr. Hino. We've not actually met, but we communicate over many years. And we may have met, I think, but, you know, a long time ago, probably. Let's continue to talk about these initiatives, and I hope that your audience has taken some warm messages from today. Thank you so much.
Thank you so much, Professor Mary. Yes, definitely. It was a wonderful discussion. We were able to discuss over the breakfast some points through which we can motivate our young girls in science. And of course, it is also very important to motivate our sons, male child as well, because they are the one who will, eventually would need to support their women, whether they are sisters, wives or whatever. With this, would like to thank Professor Ute, Professor Mary, and all the audience in the hall and virtual participants. And hopefully, I will see you all in the next event, which is scheduled on February 14th, 2023. That happened to be Valentine's Day. So, see you all again. Thank you so much for joining in. Thank you.